Usually when we think about cleaning something up, there's this expectation of efficiency. Like if you're tidying up your house, right? You pick up the trash, you put it in a garbage bag, and you just take it out.
Right, yeah, it's direct.
Exactly. It's targeted. You isolate the bad stuff, you remove it, and you leave all the good stuff completely untouched.
I mean, it makes logical sense, that's how we build things.
Right. But then you step into the world of human anatomy and look at how our bodies actually clean our blood. And suddenly, that logical, efficient trash can model it's completely thrown out the window.
Oh, absolutely. It's wild.
So today, we're taking a deep dive into the fascinating and honestly wildly misunderstood process of urine formation. We're looking at some detailed anatomy and physiology notes to understand exactly how the kidneys turn blood into urine. So, okay, let's unpack this. Imagine cleaning your house, but instead of picking up the trash, you take absolutely everything you own.
Literally everything.
Yeah, your couch, your TV, the family photos, the dog, the actual garbage. You take all of it and you throw it out onto the front lawn.
Just total unmitigated chaos.
Total chaos. And then, after dumping your entire life on the grass, you spend the rest of the day painstakingly carrying the 99% of things you actually want to keep, piece by piece, back inside the house.
Leaving just, like, the crumpled wrappers on the lawn.
Right, leaving the dust and wrappers to be hauled away. And that, incredibly, is how your kidneys work.
Yeah. And before we get into the microscopic biology of how that chaotic lawn sorting actually happens, we really have to shatter probably the single most common misconception about urine.
Oh, sure.
You will hear people say all the time that urine is, quote-unquote, just filtered blood.
Which, I mean, that sounds perfectly reasonable on the surface.
It does. It does sound reasonable, but if it were just filtered blood, you would literally be dead in a matter of hours.
Wait, really?
Yeah. The sources give us this startling statistic that really puts the scale of the kidney into perspective. The kidneys have a glomerular filtration rate of about 180 liters of fluid per day.
Okay, 180 liters. For context, for you listening, that is like 92-liter soda bottles of fluid just rushing through this system every single day.
Exactly. But think about a normal day. You are only producing about 1 to 2 liters of urine over a 24-hour period.
So wait, the math there is wild. That means your kidneys are reabsorbing roughly 99% of the fluid they initially filter out.
Yeah, 99%.
So it doesn't pick out the bad stuff. It literally filters almost everything and then aggressively takes almost all of it back.
That's the front lawn analogy perfectly applied to biology.
Well, wait, I have to stop you because I'm thinking about the sheer physical volume here. The average human, like you and me, we only have about 5 liters of blood in our entire body. How is it physically possible for the kidneys to filter 180 liters a day? Are we, like, generating new liquid from somewhere?
No, it is the exact same 5 liters.
Oh, wow.
Yeah, the kidneys are continuously, rapidly cycling your blood plasma over and over again. Your entire blood volume gets filtered something like 60 times a day.
60 times?
Yeah, it is an incredibly high-flow, high-energy system.
That is just exhausting to even think about. I mean, it seems so deeply inefficient to operate that way.
You aren't the only one to think that. And because that 99% take-back process sounds so, counterintuitive, the scientific community actually spent almost a century arguing over whether it was even real.
A century? Just arguing about how the kidney works.
Yeah, the historical drama here is fantastic because we didn't always have the technology to look at this process in action. Back in the 1840s, there was this British surgeon named Sir William Bowman, and he got his hands on some improved microscopes. He described this little capsule in the kidney, which naturally we now call Bowman's capsule, but he looked at it and assumed the whole kidney tube, the nephron, was just a secretory gland.
Meaning, he thought it actively pushed out waste, sort of like a sweat gland.
Exactly.
Like you have bad stuff in the blood, the gland secretes it directly into a tube, and boom, you get urine.
That was his theory. And honestly, it aligned perfectly with that efficient trash can logic we started with.
Right, the logical way to do it.
But then, just 2 years later, in 1844, a German physiologist named Carl Ludwig proposed a rival concept, which was the filtration reabsorption theory.
Okay.
He hypothesized that blood pressure physically forces a huge amount of fluid out first, and then the tubules must reabsorb most of it back into the blood.
So he basically predicted the front lawn analogy before anyone could actually prove it.
He did.
Okay.
It was a really elegant, mechanistically simple theory. But the massive problem was that this debate lasted for 80 years because it was completely untestable at the time.
Because it's just too small.
Right. A nephron is only a fraction of a millimeter wide. In the mid-19th century, you couldn't exactly stick a needle in there and see what the fluid was doing.
Oh, sure. They didn't have the tools.
Exactly. And what's fascinating here is how clinical observation ran way ahead of the basic science. Our sources bring up Dr. Richard Bright in the 1820s.
So even before the debate.
Right. Decades before Ludwig and Bowman are having this argument, Bright notices that if a patient has clinically significant protein in their urine, they have severe kidney disease.
So he's diagnosing the engine failure without actually knowing how the engine is built. He just knows, like protein in the urine is a massive red flag.
Exactly.
So if they couldn't see inside the nephron, how did they finally settle the debate and prove Ludwig's lawn theory was right?
It took until the 1920s. An American pharmacologist named Alfred Newton Richards pioneered a technique called micropuncture.
Micropuncture. Sounds delicate.
Extremely. He literally inserted microscopic glass pipettes by hand under a microscope into individual living nephrons in frogs.
Whoa, the steady hands you would need to hit a microscopic tube without just completely destroying it.
Unbelievable, right? And he withdrew fluid from the very beginning of the tubule and tested it. And he found it was completely protein-free, but otherwise identical in chemical composition to blood plasma.
Oh, so that proved Ludwig's initial filtration step. It's just bulk fluid.
Exactly. Then he sampled fluid further down the tubule, and the chemical composition had radically changed.
Proving the reabsorption. The body was actively taking things back as the fluid moved along.
Yep. Ludwig was right all along. You filter first, then you aggressively reabsorb.
Okay, knowing that Ludwig's mechanical theory was correct, I really want to dig into the actual mechanics of how this happens.
Yeah.
Because to filter 180 liters, you need a serious piece of biological engineering.
You really do.
So this all starts at the very beginning of the nephron with step 1, glomerular filtration.
Right. So each of your 1 million nephrons begins with the glomerulus. Think of it as a tightly wound ball of yarn. But the yarn is made of tiny, highly permeable blood vessels called capillaries.
Okay, got it.
And this entire ball is sitting inside that structure we mentioned earlier, Bowman's capsule.
And the notes mention that blood comes rushing into this ball of capillaries at a really high pressure, about 55 to 60 millimeters of mercury.
Yes.
Which, from what I understand, is unusually high for a normal capillary bed in the body. Why is the pressure spiking so intensely right there?
It is a brilliant physiological plumbing trick. The blood vessel entering the glomerulus, the afferent arteriole, is physically wider than the blood vessel leaving it, the efferent arteriole.
Oh, like putting your thumb over the end of a garden hose.
Exactly like that. You have a lot of fluid rushing in through a wide pipe trying to exit through a narrow pipe. The resulting traffic jam spikes the hydrostatic pressure.
And that pressure literally forces water and small dissolved solutes out of the blood plasma and through the capillary walls.
Right.
But those walls aren't just solid pipes, right? They are a highly specific filter. The sources describe this 3-layer membrane that the fluid has to push through.
Yeah, it's a very complex barrier.
You've got the endothelium, the basement membrane, and then these structures called podocyte foot processes that interlock to create slit diaphragms. I like to visualize this whole setup as a highly specific pasta strainer.
Well, the pasta strainer is a good next-line visual, but these 3 layers act more like a microscopic, electrically charged obstacle course.
Oh, electrically charged.
Yeah. So the first layer, the endothelium, has actual pores or windows that let fluid through.
Yeah.
The middle layer, the basement membrane, is like a dense gel that physically blocks large molecules. And those podocytes in the third layer, they literally look like microscopic feet with interlocking toes.
Okay, that's a wild image. Interlocking toes on a blood vessel.
Yeah, and the tiny gaps between those toes are the slit diaphragms.
So water, salt, glucose, and amino acids are all small enough to slip through the gaps between the toes. But your big, quote-unquote, noodles, the red blood cells and large proteins like albumin, get physically blocked and just stay in the bloodstream?
They do, but they also get electrically repelled.
Oh, right, the electrical part.
Yeah, that basement membrane is negatively charged, and most blood proteins are negatively charged too. So it's like trying to push 2 negative magnets together. The proteins literally bounce back.
Which brings us right back to Richard Bright in the 1820s. If you have clinically significant protein in your urine, it's not just some harmless quirk. It means that physical three-layer membrane is torn.
Exactly. The podocytes might be damaged, or that electrical charge is lost due to disease. That physical tear is the exact mechanism allowing those massive proteins to slip through into the urine.
So the body just used intense pressure to push 180 liters of fluid out through this microscopic negatively charged obstacle course. We've successfully thrown everything onto the front lawn.
We have.
But if the fluid just goes straight to the bladder from there, we die of severe dehydration before lunchtime. So how does the body rescue the good stuff?
That initiates the Great Reclamation Project, which is step 2, tubular reabsorption.
Right.
This newly filtered fluid, which we call filtrate, starts traveling down a long, winding tube. It passes through the proximal tubule, dives down into the loop of Henle, comes up into the distal tubule, and finally feeds into the collecting duct.
And all along this tube, the cells lining the walls are frantically grabbing the water, glucose, and sodium and just pumping them back into the bloodstream.
Yes.
And the proximal tubule seems to be doing the real heavy lifting here, right?
It really is. The proximal tubule reabsorbs about 65% of the water and sodium right off the bat.
Wow.
But crucially, under normal conditions, it takes back 100% of the filtered glucose. Your body treats glucose as premium energy. It absolutely refuses to throw it away.
I really want to understand the how here. How are these cells actually grabbing the sugar out of the fluid?
They use something called a sodium-glucose linked transporter, or SGLT.
Okay.
The cells in the tubule wall basically handcuff a sodium ion to a glucose molecule and pull them both inside together. They use the natural inward flow of sodium to drag the glucose against its concentration gradient.
That's incredibly clever.
Right.
But there's a physical limit to how fast those transporters can work, right? The notes talk about a renal threshold, which sits around 180 milligrams or deciliter of blood sugar.
Right.
So if your blood sugar spikes higher than that, the tiny cellular SGLT workers just get completely overwhelmed.
Exactly. All their transport proteins are physically occupied. They max out.
So the excess sugar just washes right past them and spills into the urine?
Yep, they just can't grab it fast enough.
This completely explains why, historically, doctors used to test for sweet urine to diagnose diabetes. The word diabetes mellitus actually refers to sweet urine.
It does.
The blood sugar was so high, the kidneys simply couldn't reabsorb it all.
And that exact mechanism is also the target of modern diabetes medications called SGLT2 inhibitors.
Wait, I've seen commercials for some of these. How do they actually work?
Drugs like empagliflozin act like a broken key in that SGLT padlock. They intentionally block the cellular workers in the proximal tubule from grabbing the glucose.
Oh.
Yeah, by deliberately inhibiting the reabsorption, you force the body to just pee the excess sugar out, which successfully lowers your overall blood sugar.
Okay, here's where it gets really interesting to me. Reclaiming salt and sugar with, cellular transporters makes complete sense. But how on earth does the kidney pull pure water back into the blood?
That's the trick.
Because you can't just grab a water molecule with a protein pump in the same way, can you?
No, you can't pump water directly. You have to trick it. And the kidney does this in the loop of Henle using a mechanism called the countercurrent multiplier.
Okay.
The loop dives deep into the center of the kidney and essentially creates a massive salt trap.
So it's manipulating the environment around the tube rather than the water itself.
Yes, exactly. The descending side of the loop lets water escape, but the ascending side is completely waterproof and aggressively pumps salt out into the surrounding kidney tissue.
So it builds an intensely salty gradient deep in the kidney.
Right.
Because water always wants to move towards salt via osmosis.
Osmosis is the absolute driving force here.
Yeah.
So later on, when the fluid reaches the final stretch called the collecting duct, the body can fine-tune exactly how much water to keep.
How does it do that?
Well, if you are dehydrated, your brain releases antidiuretic hormone, or ADH.
Okay, diuresis means making urine. So this is literally the stop making urine hormone.
Exactly. ADH travels down to the kidney and physically opens tiny water channels called aquaporins in the walls of the collecting duct.
Oh, I see.
And because the kidney tissue outside the duct is so intensely salty from the loop of Henle, the water is violently sucked out of the tubule and back into the blood.
Which concentrates the urine and saves your hydration. And the sources note that if something goes wrong and you don't produce ADH, a condition called diabetes insipidus, that collecting duct just stays locked tight.
Yes.
The water can't escape into the salty tissue, and a person can produce over 20 liters of highly dilute urine a day. You would have to drink water constantly just to survive.
It is a delicate, continuous balancing act.
Okay, so we've indiscriminately filtered the small stuff, and we've used these brilliant cellular pumps and salt gradients to rescue the water and nutrients. But wait, earlier you mentioned that massive proteins can't get through the initial filter.
Right. The large proteins stay in the blood.
What happens to a tiny toxin or a drug if it's physically glued to one of those giant proteins in the blood? Does it just stay in the body forever?
That brings us perfectly to the third step, tubular secretion.
Okay.
This is the kidney's active backup purge system. While the fluid is moving down the tubule, the cells can actively grab specific stubborn toxins, acids, or drugs from the surrounding blood capillaries and forcefully pump them directly into the tubule fluid, completely bypassing the glomerulus.
So they strip the toxin off the giant protein and manually throw it into the trash tube.
Yep, it is active transport. The cells are spending real chemical energy to pump these substances against their concentration gradients into the urine.
That's amazing.
And in the 1930s, a physiologist named Homer Smith utilized this exact mechanism. He found a substance called PAH, or para-aminohippuric acid.
Okay.
When PAH enters the kidney, it is almost 100% secreted in a single pass.
Meaning the moment blood enters the kidney, every single drop of PAH gets ripped out of the bloodstream and thrown into the tubule.
Right. Because it is so incredibly efficient, Homer Smith realized he could inject a patient with PAH, measure how much came out in the urine, and use that to calculate the exact flow rate of blood plasma through a living human kidney.
Oh, wow. No microscopic pipettes required.
Exactly.
That is brilliant. And as we're talking about active cellular pumps clearing out drugs, wait, is this how masking agents work in sports doping? Like, if an athlete takes steroids, the drugs eventually have to leave the body through this secretion step, right?
They do. Banned substances often rely heavily on tubular secretion to enter the urine to be excreted.
So if an athlete wanted to beat a urine test, they would just need to cause a traffic jam at that specific cellular pump.
Nailed it.
The notes mentioned a drug called probenecid. Does probenecid just block the pump?
It competitively inhibits the transport proteins responsible for secretion. So the cells are so busy trying to secrete the probenecid that the banned steroid is ignored.
Wow.
Yeah, the steroid stays in the blood, it never makes it into the tubule, and the athlete produces a perfectly clean urine sample.
That is wild. The World Anti-Doping Agency banning probenecid as a masking agent completely hinges on understanding the microscopic cellular biology of tubular secretion.
It does. And if we connect this to the bigger picture,
Right.
we really have to rethink the entire purpose of the kidney.
How so?
Well, we started this deep dive talking about a trash can, but waste removal, getting rid of urea and drug metabolites is honestly just a side gig for these organs.
A side gig. We literally just spent all this time talking about filtering out toxins.
We did, but urine formation is ultimately how your body achieves homeostasis. It is the master regulator.
Okay.
When the tubule secretes hydrogen ions and reabsorbs bicarbonate, it is actively regulating your blood's acid-base balance, keeping your pH in a strict survival range.
Right.
When it reabsorbs sodium and water, it's regulating your total blood volume, which directly dictates your blood pressure.
So the kidneys aren't just a garbage disposal. They are the control room for the entire cardiovascular system.
They are, and they function as a vital endocrine organ too.
Really?
Yeah, the kidneys don't just filter, they produce hormones. They secrete renin, which triggers a cascade to constrict blood vessels and raise blood pressure. They produce erythropoietin, or EPO, which travels to your bone marrow and commands it to produce more red blood cells. They even convert vitamin D into its active form so your intestines can actually absorb calcium.
The causality there is amazing, because if your kidneys fail, you don't just build up toxins, you become anemic because your bones stop getting the EPO signal to make blood.
Exactly.
You develop bone density issues because the vitamin D isn't activated. It is all deeply, deeply interconnected.
And when you start looking at the world through this physiological lens, so much of everyday life just makes sense.
Give me an example.
Take alcohol. Why does drinking alcohol make you have to urinate so frequently?
Oh, because it messes with that ADH hormone we talked about.
Exactly. Alcohol physically suppresses the release of antidiuretic hormone from your brain.
So without ADH.
Without ADH, the aquaporins in your collecting duct lock up. You can't reabsorb the water into that salty tissue, and you rapidly lose fluid, which leads directly to the severe dehydration associated with a hangover.
That makes perfect sense. Or think about why endurance athletes drink sports drinks packed with sodium and potassium instead of just plain water.
Yes.
They are giving the proximal tubule and the loop of Henle the exact osmotic tools they need to pull fluid back into the blood and maintain that blood volume.
Absolutely. And the applications in modern medicine are staggering.
Mm-hmm.
For anyone who knows someone on blood pressure medication, which is millions of people globally, almost all of those drugs work by tweaking one tiny mechanism in this three-step urine formation process.
Really? Like diuretics.
Right. Diuretics might block the sodium transporters in the loop of Henle, meaning salt stays in the tubule, water stays with the salt, and you pee out excess fluid, lowering your overall blood volume.
And ACE inhibitors.
ACE inhibitors interfere with the hormonal signals that tell the kidney to reabsorb sodium and constrict blood vessels. The entire field of modern hypertension management rests on knowing exactly which cellular pump or hormone receptor in the nephron a specific drug targets.
So what does this all mean? We started with 1 million microscopic, negatively charged obstacle courses. We talked about how hydrostatic pressure forces a massive amount of fluid out in glomerular filtration.
Right, generating an unbelievable 180 liters of filtrate a day.
Followed by tubular reabsorption, where the body uses cellular pumps like SGLT2 and the brilliant salt trap of loop of Henle to frantically haul 99% of that water, salt, and sugar back into the bloodstream.
Yep.
And finally, tubular secretion, where the body actively burns energy to manually pump those stubborn protein-bound toxins straight from the capillaries into the final urine output.
Resulting in just 1 to 2 liters of highly processed, meticulously calibrated fluid.
It is a marvel of continuous minute-by-minute regulation.
It really is.
It really is. And it leaves me with this one final thought, something to kind of chew on after you finish listening today. From a pure engineering standpoint, the kidney's design, throwing almost everything away just to spend massive amounts of biological energy hauling 99% of it back, seems incredibly inefficient.
It does.
But when you look at how tightly it regulates our blood pressure, our acid levels, our hydration, maybe evolution doesn't care about simple mechanical efficiency.
That's a great point.
Maybe evolution values extreme minute-by-minute adaptability. By filtering everything constantly, the body can make instantaneous adjustments to anything that changes in your internal environment. It gives you total control.
It might look like chaos on the front lawn, but it is the exact reason you are alive and balanced right now.
Thank you so much for joining us on this deep dive.
Thank you for listening. Keep asking questions.